Hello, tonight we're going to talk about DNA technology. DNA technology involves all sorts of techniques used for gene therapy, for changing the genetics of various organisms. For example, with this picture you see in front of you, this is a tobacco plant, and they inserted a gene from a firefly into the tobacco plant and obviously you can see the tobacco plant now glows in the dark. They did this to determine if it was possible to move genes from one type of organism into another type of organism, and in this case from an animal into a plant. Very, very different organisms, and they did. So this is DNA technology. Some of the terminology that you'll need to be familiar with will first of all be recombinant DNA. This is any DNA that has new DNA added to it, whether it's from the same organism or from other organisms. Genetic engineering is the direct manipulation of genes for some purpose. We have all sorts of things that we have genetically engineered. We'll get into those. Biotechnology is the manipulation of living organisms or their components to produce useful products. For example, we have bacteria that produce human insulin for us, and then diabetics can have insulin. Another term is a vector. A vector is what you use to move one gene from one organism to another organism, and the two usual vectors are viruses or plasmids. So let's talk about recombinant DNA and cloning. Now, we already talked about Dolly the sheep and how they cloned her and some of the issues that they had with that. She was the first mammal that had ever been cloned and the first organism that had telomeres that was ever cloned. So what do we do now? How have we changed in the last 15, 20 years so that what we're doing is different? Okay, the first thing they have to do is you have to isolate a plasmid or other vector, a virus, that you're going to use to insert the genes with. Okay, now we're going to talk about bacteria because it's easy to work with bacteria. Bacteria only have one chromosome. It's a circular DNA, and it's fairly easy to get bacteria to change genetically. Okay, so a plasmid is a circular piece of DNA, and what you're going to do then is to use a restriction enzyme. Now, restriction enzymes are enzymes that cut DNA at a specific sequence. So, for example, ATTCCG or something like that. Whenever the restriction enzyme comes across that 6-letter sequence, it's going to cut it. Okay, and then you use the exact same restriction enzyme and cut the needed gene out of whatever organism you're going to use it from. Let's say a glow-in-the-dark gene. And then you use the enzyme DNA ligase to glue the two pieces that you have now cut together. Now, the reason you use the same restriction enzyme is because then you will end up with the exact same letter combination, ATTCCG. Okay, so here's what happens. Here's the enzyme restriction site that we like. Okay, this particular enzyme is going to use this one. By the way, there are lots of different restriction enzymes. Many viruses have them. That's where we've isolated them from. Okay, so it's going to come in and it's going to cut at that sequence. Now, when it does, it leaves a little bit of the DNA hanging out on both sides. It's called sticky ends. I don't know who named it that, but I think it's kind of funny. You take a DNA fragment with the same restriction enzyme that you've cut it. Okay, it has the same sequence there, and you're going to then put it together, and then ligase will come in and glue those two parts that are broken apart together, and now you have recombinant DNA. Now, if you go back, I made a little thing here in red. it has to be cDNA, which is complementary DNA. The reason is, if you take a human gene for, let's say, insulin, and you put it inside a bacteria, if there are any introns in that DNA, that bacteria cannot copy the gene, cannot make insulin, because bacteria are incapable, they do not have spliceosomes, they are incapable of cutting out the introns. So you have to have the regular complementary DNA. So how they create that is kind of another issue. What they do is they take the mature messenger RNA and use an enzyme called reverse transcriptase, which changes the messenger RNA back into DNA, and that DNA is now called cDNA. That's the DNA you are going to put inside the bacteria. Okay, then they add another gene to your plasmid or virus. It's called a reporting gene. You're going to attach it to the cDNA, doing the exact same restriction enzyme stuff. This gene is going to do something to make it obvious that the bacteria has it, like glow in the dark or ampicillin resistance or something, so that this will survive. That I'll know this one bacteria has the gene that I want it to have. And once I know that, then I can culture that bacteria and grow that one, and it'll produce whatever product that I wanted it to produce, like the insulin or growth hormone or lots of different things that we have bacteria produce for us. Okay, so once we've got that DNA and I've got the reporter genes attached to it, you go to the plasmid, cut the plasmid with the same restriction enzyme, it inserts, now we've got the plasmid. Now we have to get this plasmid inside the bacteria. Okay, that's what ligase does, just glues it back together. Okay, we're going to put that plasmid back inside the bacteria by a process called transformation, and that's going to be on the next slide. That process of transformation is a lab we're actually going to be doing in class. That bacteria is going to grow, and I know it's going to have it, and I can use that bacteria. I could put lots of genes inside that bacteria to make it do lots of things for me. Some of the things we currently do with bacteria. We have bacteria that we've added a gene to them which allows them to eat oil. So we can put them on oil spills, and they eat the oil and clean up the environment. We can also put a lifespan into them so that after they divide X number of times, then they die. We can take bacteria and make them produce insulin, bacteria to produce proteins that break down blood clots, and the list goes on and on and on. We use this type of technology more than you're aware of. Okay, so let's look at this bacterial transformation, because this is the lab we're going to be doing. It's real important that we understand what's happening. Okay, first thing we're going to do is add some calcium chloride to the bacteria. What that does is it makes the plasma membrane very permeable. It almost pokes holes in it. And then we add the plasmid solution surrounding the bacteria. Then you put the bacteria on ice. What that does is it causes the bacteria to kind of shrink a little bit. And then you put it in 40°C water, which is going to heat shock it. When it does, it rapidly expands. The plasma membrane rapidly expands. Hopefully, in that surrounding water that it sucked in so it could expand, some plasmids got sucked in. Then you put the bacteria back on ice, and then you put it on a petri dish and you grow it. And if the bacteria grow, then you know that it had the plasmid that we added to it. Now, you remember that reporter gene? We're going to grow bacteria on ampicillin plates. That means plates with an antibiotic on them. We're going to put a reporter gene in there which will allow the bacteria to be resistant to ampicillin. So any bacteria that grow on the ampicillin plates will have the plasmid, and we will know exactly which ones they are. There are other, like I said, other reporter genes. It can be ones that make it glow, ones that make it turn blue or green, various things. Okay, we're going to watch a little video now about electrophoresis. This is another technique that is very important that you understand how it works. This is used all the time, and this is almost like old technology now. It's so frequently used. Okay, so here we go.
To first make the agarose gel, we add powdered agarose to 1x TBE buffer. We'll then mix the solution as best we can and heat it to boiling in a microwave. We will monitor the mixture closely as it sits in the microwave to ensure that it doesn't burn. Once the mixture begins to boil, we'll remove it repeatedly from the microwave and agitate it gently to ensure that all of the agarose has been mixed with the TBE buffer. Once the agarose has been mixed into the TBE buffer, we're ready to pour our gel. We start by assembling our gel apparatus with a comb, a casing, and 2 barricades. We then pour the hot agarose into the gel casing and allow it to sit at room temperature until it solidifies. Once the agarose solidifies, we cover the gel with TBE buffer and then remove the comb. Removing the comb reveals wells, which are cavities in the agarose into which we can place our DNA samples. We'll use a micropipetter and plastic pipette tips that are sterile and free of DNases to aliquot a small quantity of each of our samples into their respective wells. The samples have been mixed with a blue dye, which will help us monitor how far they've moved in the gel when we place an electrical current through it. The same procedure is used to place each sample into its respective well until all samples have been placed in the gel. At this point in time, we're now ready to connect our gel to a power source to place an electrical current through it that will move the DNA through the gel. We set the voltage to 100 volts and press the running man to make it run. When we do this, we actually see bubbles arise from one of the leads, letting us know that there is electricity passing through the TBE buffer and therefore through the gel. As time proceeds, we'll notice that the dye actually moves through the gel, giving us a very distinct front that we can follow. Notice there are 2 fronts here, an aqua-colored one and a blue one. At this point in time, we're now prepared to analyze our gel. There are 2 methods that help us analyze gels. The first utilizes what's known as an intercalating agent, which binds to DNA and fluoresces under ultraviolet light. This intercalating agent was added to the agarose TBE mixture before we poured the gel. The gel shown here contains an intercalating reagent and is exposed to ultraviolet light, and as can be seen, the bands representing fragments of DNA fluoresce a bright green. The second method that we can use to help us analyze the gel is one where we add a stain or dye to the gel, which thereby stains or dyes the DNA, allowing us to visualize the bands. We start by placing our gel into a container and pouring the dye on it. We let it incubate with the dye for 1 minute before pouring the dye back into its container. After removing the dye, we now must destain the gel by first adding water and then placing the gel into the 40-degree incubator for set periods of time. Once the water in the gel container becomes too blue, we'll pour it off, add more water, and repeat until the gel efficiently destains. Once the gel has fully destained, we can then begin to visualize the bands of DNA, similar to the way we were able to visualize them with the intercalating agent.
Okay, another thing that we do, another technique, is called polymerase chain reaction. A polymerase chain reaction is when we're going to make millions and millions of copies of a small segment of DNA. They do this because on that gel electrophoresis, you need to have enough DNA so that you can see it when you stain it or when you intercalate it. Okay, so you need to have lots of copies of it. So you go through a process called PCR. Basically, what happens with PCR is that you take a sequence of DNA, and through a series of reactions, the DNA is going to be split, primers are added, it's copied, and then those will be split again and copied again, and it exponentially divides, and you end up with millions and millions of copies. 2 copies, 4 copies, 8, 16, and so on and so forth, exponentially. Okay, that's the process. Now, why would they do that? Well, think about this. If you were at a crime scene and there was one tiny drop of blood left by the criminal, well, in the blood, the only cells that actually have DNA are white blood cells. So you have to find one white blood cell, or 2, or however many you can find within that blood sample. And then you have to take that DNA, which is so tiny, you have to multiply it. You have to copy it. And so they go through PCR and make millions and millions of copies of it so that when you put it in the electrophoresis, you actually can see it. So very tiny samples of blood or samples of anything are enough to give you enough DNA so you can actually visualize it. Okay, so let's go back through that electrophoresis so you can see exactly, again, what that person on the video told you that they did. Okay? So first what they're going to do is load, in this case this gel is an upright gel as opposed to one that was laying down, but they're basically the same. You load the gel into the wells, the DNA samples in here. You turn it on with electricity, and then the DNA is negatively charged, and so it's going to run towards the positive pole. Positives and negatives attract each other. The larger molecules go very slow. The gel that it's running through is very much like spider web material, and if you have a very large piece of DNA, it has to work its way between all the webbing to get through the DNA, or to get through the gel. If it's a small piece of DNA, it doesn't have as much of a trail or tail behind it that it's pulling to get through the little spider web stuff. So as a result, small fragments move very fast, large fragments move very, very slow. And so it separates by size. These are real small, so they're going to separate like this. Okay? Now, just looking at that, do you see any 2 samples that are identical? Well, this one right here, let's say, was from the crime scene. Each one of these is somebody else. This one and this one. This is the guy who did it, who had blood at the crime scene. Okay, so you can see that. Okay, that leads to another term that we need to be aware of. It's called a restriction fragment length polymorphism, or RFLPs. Each of those little sections, each of those little segments of DNA is called an RFLP. When you use that restriction enzyme, it's going to cut the DNA at that sequence wherever it is, in the middle of genes and interons, wherever it is. It's going to cut the DNA. Because everybody in the world has mutations, the length of those restriction fragment length polymorphisms is going to be slightly different. Even if you're 1 letter off, but if it happens to be at that restriction enzyme recognition site, then it's not going to cut it there. So you're going to have where it might cut it on somebody else, it might not cut it on you. And so you get different lengths because of your mutations as opposed to somebody else. And even those little slight differences Because the DNA is so huge, those slight differences are enough to be able to determine who did the crime or who the father is, who the paternity is for a certain child. So that's what it's all about. Okay, so again, this is looking at an electrophoresis type of thing. Now, I mentioned earlier that bacteria cannot code a gene with introns, and so you have to be able to use cDNA, complementary DNA. So you use this enzyme right here, reverse transcriptase, to change messenger RNA back into DNA without introns. You want to use mature messenger RNA. So you have to have complementary DNA to be able to work with the bacteria. Now, here are some of the things that we currently do with DNA, some of the uses of the technology. It's awesome. We have genes for pest resistance inserted in the plants, so pests won't eat the plants. You can have genes that clean up oil spills, and so bacteria cleaning up oil spills. We've got bacteria that are producing genes for being able to dissolve blood clots. So if you have a heart attack, you don't die. Growth hormone, many, many, many others. Back, more than 20 years ago now, scientists went through a project called the Human Genome Project. They wanted to find out what the actual sequence of letters were in a human chromosome and what all the genes were. But the first thing they had to find out is what the letters were. What are the nucleotides sequence? So they picked one person, and I don't know who that person was, but it'd be really cool to know. They picked one person's DNA, and then they had to figure out how to figure out this order of ATT, CAT, AGG, that order for all 46 of their chromosomes. It took them 15 years to figure out how to do it, and once they figured out how to do it, then they were able to decode all 46 chromosomes in less than a year and a half. I think that's pretty cool. So now they know the correct order of letters for that one person. Since then, and that happened just after 2000. Since then, they've been trying to figure out what every gene is used for. And they're making quite a bit of headway with that. What they want to be able to do is to be able to locate where the bad genes are in a person who might have a genetic disease, be able to replace them with good genes. So using gene therapy, using a virus or something, insert the correct genes and be able to cut out the bad genes so a person might never have diabetes again, or whatever other genetic diseases it might be. And that has led to some interesting techniques that have occurred since then. The development of transgenic organisms, and genetically modified food. So what they have done is a transgenic organism is an organism that has DNA altered from another species. So remember that tobacco plant that had the glow-in-the-dark firefly genes in it? That's a transgenic organism. Other ones that we currently have, something called yellow rice. Yellow rice is a rice that we added genes to produce, I think it's vitamin E, so that people in poor countries who don't get enough vitamin E will get enough vitamin E, and that prevents blindness. However, because it's yellow, the people in those countries don't necessarily like the rice. It doesn't change the flavor, but it's just different, and so they're not eating it. But they wouldn't go blind if they ate it. Well, I think I'd eat it. Plants that won't spoil as quickly because we've added genes to them that prevent them from spoiling. Tobacco plants that are now growing antibiotics for us. That's pretty cool. Transgenic animals that we currently have, cows that can excrete human hormones through their milk. Yeah, that's kind of an interesting one. Here are some others. How'd you like to have a pet glow-in-the-dark organism? Well, this pig definitely is a glow-in-the-dark, and this is another pig right next to it. They added genes to it from actually I think it's an algae that glows, a bioluminescent algae, and so that pig now bioluminesces. This rat over here bioluminesces, but because the fur covers it, you can only see it in areas where the fur is not. This one I think is kind of fake. Sorry, you don't get a bunny like that. But they do have glow-in-the-dark monkeys and beagles. They've changed fish. The neon fish that you can buy at Walmart are actually transgenic organisms. So there's some kind of cool things going on there. Genetically modified are organisms that have DNA from the same species. So if we found a person who had diabetes, who could not produce insulin, if we were able to genetically cut out their bad insulin gene and insert a good insulin gene, that person would be genetically modified. Okay, so some organisms that we currently have genetically modified, not humans, but ones that we have. Salmon receiving more genes for growth hormone from other salmon so that they grow larger. Off the top of my head, I can't think of others, but there's several. We're going to watch a little YouTube now.
Well, I just heard the news today. It seems my life is gonna change. I close my eyes, begin to pray, then tears of joy stream down my face where lungs wide open.
As you saw from the video, there's a lot of research that's going into gene therapies, and they're trying to cure a lot of diseases, like they said, right at the source. What I would like you to do tonight is to research one transgenic or genetically modified organism and write a short essay describing what organism it is and what type of gene therapy went through to create that type of organism. You know, how'd they create it? Where did the genes come from to make this transgenic organism? You can use a glow in the dark. You can use the neon fish. You could use lots of stuff. Just Google transgenic organism or genetically modified and see what you can come up with. Okay? That's your homework for tonight. Have a nice night.